Thanks so much for having me. Pleasure to pitch a little project of ours here. Thanks for the introduction. Yeah, I'm with Kurt-Schwabe-Institute and also with TU Dresden, and we are interested in automating biochemical processes at the Kurt-Schwabe-Institute, always in the context of all kinds of sensor technologies. Now, let me show you what we actually do at the KSI. So you can see here a couple of application areas that we try to cover. It goes from animal welfare, so we're interested in the health of cows and pigs and whatnot, through process control in the context of digital society and industry, but also a lot of environmental sensing. So we're making especially electrochemical and gas sensors to study, for instance, contaminants in soil or water, heavy metals, for instance. And one of the projects that we have recently been very active in are biomedical nanotechnologies and bioanalytical technologies in the context of drug discovery, but also diagnostics. Yeah. And this is what the project is about that I'd like to speak about very briefly today. Just because you probably don't know where the KSI is, we're kind of in the middle of the Bermuda Triangle between Dresden, Leipzig, and Chemnitz. So if you happen to pass by, it's a lovely area. Please stop by and stop in for a coffee. Happy to have you. So what we're doing is we're trying to make enabling technologies to study molecular interactions. So, and by that we mean quite basically understanding how molecules of any kind, that could be proteins, DNA, peptides, small molecules, doesn't matter too much, but how they interact, how they bind, how long they stay bound. And that could be a simple bimolecular interaction, but it could also be, for instance, a ternary complex between A, B, and C. And so what we're trying to extract from these experiments are rate constants and affinity constants, to understand and tailor and engineer how these interactions work. And the outcome of these studies and the benefit of the instruments and the sensors that we make is in these areas. So we're trying to develop enabling tools that help others to engineer smart drugs. We see a bispecific antibody as an example here, but that could be also small molecules or others, in order to treat complex diseases like, for instance, cancer. And so in part of this journey, we have started recently a project where we are developing a sensor-enabled chembot that should also be AI-powered. So the idea is here that we make an automated system that uses sensors to characterize raw materials before it actually runs reactions in an automated fashion on raw materials. Again, this could be biomolecules like antibodies, could be fluorescent dyes, could be DNA molecules. And the idea is that the way that these reactions are being run is designed with AI-assisted processes and then run automatically by a liquid handling system that is combined with sensors, with integrated sensors that can actually analyze molecules before they start a reaction. And then at the end of the reaction, we also want to purify what has been produced and analyze that, and in the end aliquot it. So this is not, as we've heard in the previous talk, this is not intended for large-scale bioreactors, just the opposite. We're addressing small-scale reactions that are being run in all kinds of academic labs or also pharma and biotechnological labs worldwide. So this is a bit the project. We're trying to make a chembot, and if you're interested in robotics, in system integration, in automation, programming of any kind, or process control, then please reach out. We'd be happy to have a chat. And we're looking to either hire or collaborate with engineers and scientists in that field who like systems, who like to integrate stuff and build instruments and biochips for analysis of biomolecules. Thanks so much.